You know, if you were to open up your smartphone right now and just look at your text messages, you'd probably see this like distinct hierarchy of communication.
Oh, definitely.
You've got those massive multi-paragraph texts you send when things get deeply emotional or complicated.
Right, the novels.
Exactly, the novels. And then you have the short, highly functional text, right? Like on my way or buy milk or call
Yeah,
me.
the quick utility messaging.
Right. And we always hear about DNA being this grand blueprint of life and massive proteins being the heavy machinery doing all the physical labor.
Mm.
But beneath all of that, there is this completely invisible, hyper-fast network of cellular text messages just quietly running the show in the background.
It really is a staggering biological network.
Yeah.
And it flies under the radar primarily because, well, these messengers are physically minuscule.
Tiny.
Right. But when you actually look at the raw data of cellular function, I mean, their influence dictates almost everything keeping you alive right now.
Which is exactly why we're doing this. Welcome to today's deep dive. Today, we are exploring a massive body of research and scientific literature all about peptides.
Yes, the biological middle children.
The middle children, bridging the gap between, basic single amino acids and those massive complex proteins. And our mission today is really to demystify these tiny chains.
And to uncover why they're mediating between 15% and 40% of all protein-to-protein interactions in human cells.
Wait, 40%? That is a huge chunk of our biology.
It's massive. And they are so effective that they actually make up at least 10% of the entire global pharmaceutical market right now.
Wow. Okay, so to understand how they control so much of our biology, we should probably establish exactly what a peptide is, right? Like where it sits on the biological sizing chart.
Let's start with the basic building block, which is the amino acid.
Okay.
If an amino acid is a single letter of the alphabet, a peptide is a short word or maybe a brief sentence.
Got it.
Strictly defined by the literature, it's a chain of anywhere from 2 to 50 amino acids held together by peptide bonds.
Just 2 to 50.
Right. And they usually take a linear shape.
Yeah.
So they have an apipine group at the front end, which we call the N-terminal, and a carboxyl group at the back end, the C-terminal.
Okay, and they have all these specific names based on length, right? Like monopeptide for 1, dipeptide for 2, up to undecayed peptide for 11, and anything under 20 is an oligopeptide.
Exactly. You're just counting the links in the chain.
But I want to push back a little on that 2 to 50 cutoff, because the literature highlights this specific threshold, noting that a polypeptide, which is just a longer continuous chain, only officially becomes a protein when it hits a molecular mass of 10,000 Daltons.
Which roughly translates to about 50 amino acids.
Right. So my question is a 51 amino acid chain really a completely different biological beast than a 49 amino acid chain? I mean, that 10,000 Dalton cutoff feels incredibly arbitrary.
Like a convenient human label.
Yeah, exactly. Just slapped onto a continuous spectrum so we can organize our textbooks.
Well, it's funny because the physics actually back up your skepticism.
Yeah.
I mean, nature obviously doesn't count to 50 and throw a switch.
Right.
However, that molecular mass threshold, it does represent a genuine tipping point in physical behavior.
How so?
Well, when a chain is short, it remains linear. It's flexible, it's agile.
Like a text message.
Exactly. It can zip through the bloodstream, bind to a highly specific cellular receptor, deliver its instructional message, and then be rapidly broken down to prevent, overstimulation.
So the small size is really all about agility and energy efficiency.
Absolutely.
Yeah.
But once a chain crosses that 10,000 Dalton threshold, it simply has too much mass and too much internal chemistry to stay linear.
It gets too heavy.
Right. The varying chemical charges of all those dozens of amino acids start interacting with each other, and the chain literally collapses in on itself.
Oh.
Yeah, it folds into complex three-dimensional shapes. So it stops being a nimble, flexible messenger and starts taking on structural, mechanical, or enzymatic roles.
It becomes the heavy machinery.
Yes, it becomes a true protein. So the transition from agile messenger to heavy machinery is very real, even if the exact number 50 is just our way of categorizing it.
That makes total sense. Which means, I assume, the way the body manufactures these agile text messages must be totally different from how it builds the folded, heavy machinery.
There are a few different ways, yeah.
So let's look at the actual assembly process, starting with the standard biological machinery, the ribosome.
Right. The ribosome operates basically like a molecular ticker tape machine.
Okay.
Your DNA sends a genetic blueprint out to the ribosome, which reads the code sequentially. It pulls in the corresponding amino acids, links them together, and creates what we call a ribosomal peptide.
Standard 3D printing, basically.
But the curious thing is that the ribosome doesn't just print out the final active peptide.
It doesn't.
No, it usually prints a much longer, inactive precursor draft. In the literature, this is known as a propeptide, or a proprotein.
Wait, if the body prints a massive precursor just to chop it down into a smaller active peptide later, there has to be a functional reason for the extra work, right?
There is.
It sounds like a biological safety catch. I mean, if you manufacture a highly potent hormone in one part of the cell, you probably don't want it randomly activating before it reaches its actual destination.
You definitely do not.
So you print it with a safety cap, ship it out, and then enzymes cleave the cap off only when it arrives. And the source calls that cleavage process proteolysis, right?
Yes, proteolysis.
Yeah.
And you've hit on a major evolutionary advantage there, because it allows the cell to store vast quantities of a signaling molecule in an inactive state,
Just ready to go.
ready to be deployed instantly. When a stressor hits, the cell doesn't have to wait for the ribosome to read the DNA and print new molecules from scratch.
That'd take way too long.
Exactly. It just releases enzymes to snip the propeptides, flooding the system with active messengers in milliseconds.
Okay, so that's the standard genetic ticker tape method. But the research also details a secondary, completely rogue manufacturing system.
Yes. Non-ribosomal peptides.
Right. These totally bypass the standard ribosome ticker tape.
They do. And you'll find this alternative system primarily in unicellular organisms, plants and fungi.
Okay.
Instead of a ribosome reading a strict genetic code, these organisms utilize these massive modular enzyme complexes called non-ribosomal peptide synthetases.
So if the ribosome is a highly regulated, automated assembly line, these synthetases act more like underground artisan workshops.
This is a great way to put it.
Because they aren't bound by the strict rules of genetic coding, which means they can weld totally different chemical materials together.
No.
But exactly what kinds of materials are we talking about here?
Well, because these synthetases actually share evolutionary roots with the machinery that builds fatty acids, they frequently churn out bizarre hybrid compounds. They'll fuse peptides directly to lipids, or they'll incorporate strange, rigid chemical rings like oxazoles and thiazoles right into the chain.
The obvious question then is the evolutionary benefit. I mean, a microbe fighting for resources in the dirt doesn't have the luxury of wasting energy.
No, not at all.
So why run a complex artisan workshop to build these hybrid peptides? My assumption is that standard linear ribosomal peptides are simply too fragile.
You're on the right track.
They have loose N-terminal and C-terminal ends, which makes it incredibly easy for an enemy microbe's enzymes to just grab them and fray them apart.
That is the core of the strategy, yes. Survival in the microbial world is raw chemical warfare.
Yeah.
To defend territory, bacteria and fungi need weapons that won't degrade in harsh environments.
Okay.
So using this non-ribosomal method, they can actually build cyclic peptides.
Cyclic, so they loop them.
Exactly. They attach the head of the chain directly to the tail, forming a closed loop. Because there are no loose ends, enemy enzymes can't easily grip or cleave them.
Wow, it's literally a biological circle the wagons approach.
It really is. And these microbes use this artisan method to mass produce powerful degradation-resistant antibiotics, things like microcins and bacteriocins, to ruthlessly eliminate competitors.
Brutal.
Yeah. Even glutathione, which is a critical antioxidant used across multiple species to defend against severe cellular damage, is constructed in these specialized enzyme workshops.
Man, the idea that biology modifies linear chains into closed loops for extreme survival naturally leads us to how larger animals manipulate peptides for their own extreme purposes.
Right, post-translational modifications.
Yes, modifications are everywhere. I mean, cells might add a phosphate group to alter a charge or attach sulfur, or even loop the peptide into a microscopic lasso structure, which the text called a lariat.
Yes, a lariat structure.
But out of all the modifications detailed in the literature, the one found in platypus venom is absolutely wild.
Oh, the racistization of L-amino acids to D-amino acids.
Yes.
It is a stunning piece of evolutionary chemistry.
Let's break this down for the listener, because a platypus is already an anomaly, right? It's got a duckbill, it lays eggs, and it has venomous ankle spurs.
It's a very strange creature.
Very strange. But its venom operates on this mind-bending chemical mirror trick. So if almost all biological life on Earth utilizes L-amino acids, which means they are physically structured in a left-handed geometry, then our entire biological defense system is built to process left-handed threats.
Right. Think of it like this. Every receptor, every clearing enzyme in your body is essentially shaped like a left-handed catcher's mitt.
Okay.
It's designed perfectly to catch and dismantle left-handed L-amino acids.
So when the platypus venom gland deploys an enzyme that actively flips a specific amino acid in the venom chain into a D-amino acid, which is a right-handed mirror image, it is basically creating an undetectable stealth weapon.
Exactly. When that mirrored venom hits a victim's bloodstream, our left-handed catcher's mitts simply cannot catch it.
Because it's backwards.
Right. They can't even recognize it as a biological molecule. The geometric flip means the victim's natural clearing enzymes just slide right off the venom peptide.
Oh, man.
And because it cannot be broken down by the standard biological defense grid, the venom remains active in the nervous system for incredibly long periods. It causes severe, sustained pain that traditional painkillers really struggle to touch.
Nature weaponizing molecular geometry. It's brilliant and terrifying.
It truly is.
Now, keeping on the theme of breaking proteins down, the research details another highly modified category called peptones. And unlike venom, these aren't weaponized.
No, peptones are very different.
They are created through aggressive proteolysis or digestion of animal meat or milk.
Right. Peptones are essentially a highly concentrated, nutrient-dense soup of small peptides, leftover fats, salts, and vitamins derived from breaking down complex animal proteins.
And the source mentions they are utilized almost universally in laboratories as the primary nutrient medium to cultivate bacteria and fungi.
That's right, it's lab food.
But with all our modern precision biotechnology, why are we feeding partially digested animal meat to lab bacteria? I mean, you'd think it would be cleaner and more controlled to synthesize a perfectly calibrated bacterial food entirely from scratch in a lab.
You would think so, but it comes down to the energetic cost of biological assembly.
Okay, how so?
Think of a bacterium as a mechanic trying to build a fleet of cars.
Yeah.
If you provide them with perfectly pure, synthesized raw iron and carbon, they have to smelt the steel, forge the parts, and then assemble the car.
That takes massive amounts of time and energy.
Exactly.
So giving them peptones is like, it's like giving them a junkyard of slightly dismantled, salvaged cars.
That is the perfect analogy, yes. Natural proteolysis of meat doesn't reduce the protein all the way down to single amino acid dust.
It leaves chunks.
Right, it chops it into highly accessible pre-assembled chunks, oligopeptides that are 2, 3, or maybe 4 amino acids long.
Oh, I see.
And the bacteria have evolved specialized transport channels on their surface designed to literally inhale these pre-assembled chunks whole. They immediately slot these ready-made biological tools into their own machinery.
So nature's messy recycling process is just way faster.
Far more efficient. It provides a fast-acting toolkit that a sterile, purely synthetic design really struggles to beat.
Man, that's fascinating. All right, so we've covered how microbes build hybrid weapons, how venom uses mirrored geometry, and how lab bacteria feast on salvaged peptide parts.
Right.
Let's bring this directly to human biology, because you mentioned earlier that up to 40% of cell interactions are governed by these chains. How exactly is the human body orchestrating all this traffic?
Well, the body organizes this vast communication network into specific functional families based on, where they operate and what they attach to.
Okay.
For instance, you have neuropeptides, which are highly active in neural tissue and govern brain function. You have peptide hormones, which act as systemic regulators traveling through the bloodstream. And you have lipopeptides, which feature a fat molecule, covalently attached to the peptide chain.
And the literature specifically zeroes in on a class of lipopeptides called peptidins and their interaction with GPCRs.
Yes. GPCRs, or G-protein-coupled receptors, basically act as the critical sensory antenna for your cells.
They just sit on the surface waiting for external signals.
Exactly. And peptidins are uniquely engineered by the body to exploit the cell membrane itself.
Well, because the cell membrane is made of lipids, right? And the peptidin has a lipid tail attached to it, so I'm guessing it uses that tail as an anchor.
You're exactly right.
So instead of just bumping into the antenna from the outside and floating away, the peptidin physically wedges its lipid tail into the membrane wall, locking itself right next to the GPCR.
Yes, it docks itself.
And then it can just continually jam or modulate the antenna's signal from the inside out.
You've perfectly described the mechanism. By anchoring into that lipid bilayer, peptidins don't wash away in the extracellular fluid. They stay put, which allows for highly prolonged, localized control over how that specific cell reacts to its environment.
It's an incredibly targeted system, and when you look closely at these specific peptide families, you start to realize how deeply they govern our daily human experience,
Oh, absolutely. Look at the opioid peptide family, for instance.
which includes your endorphins and
Look,
enkephalins.
right. These dictate our baseline sense of pain relief, euphoria, and stress response. And what's deeply fascinating about the opioid family is how the body synthesizes them.
How so?
They are largely derived from a single massive propeptide precursor called POMC.
Wait, POMC. So instead of printing 10 different pain relief text messages from scratch, the cell prints one massive POMC molecule and then chops it up to release multiple different endorphins simultaneously.
Yeah, it's a complete masterstroke of biological efficiency.
That is so smart. And then you have the tachykinin family, which operates on kind of the other end of the spectrum, right?
Right, the pain side.
Yeah, it includes substance P, which is an undecayed peptide 11 amino acids long that is heavily responsible for transmitting intense pain signals and triggering localized inflammation.
And beyond pain, you've got antimicrobial families built right into our epithelial immune defenses, like magnins.
Oh, and pancreatic peptides, right, such as neuropeptide Y, which profoundly drives appetite, carbohydrate cravings, and even fat storage.
Exactly. And beyond those systemic controllers, the research outlines some highly specific outliers that function essentially as biological distress flares.
Distress flares, like what?
BNP is a perfect example. B-type natriuretic peptide. It isn't synthesized in the brain or the gut. It's produced directly within the myocardium, the muscular wall of the heart.
So when the heart muscle experiences dangerous levels of physical stretching, like from high blood pressure or volume overload, it immediately secretes BNP into the bloodstream.
Yes.
And it acts as both a structural distress signal and a physiological mechanism, right? Doesn't it force the kidneys to excrete salt and water to lower blood pressure and save the heart?
It does exactly that. And because it's such a reliable, specific signal, modern medicine relies heavily on measuring blood BNP levels to diagnose the severity of heart failure.
That's incredible.
We also see external peptides profoundly interacting with our biology.
Mm.
For example, gelane is a peptide isolated from the royal jelly of honeybees that shows potent antibacterial properties.
Royal jelly, really?
Yeah. And there's a traditional Chinese medicine known as Kola Kori Asini, derived from donkey hide.
Donkey hide.
Yes. And it contains specific peptidic components that research indicates actively stimulate hematopoiesis, which is the physiological formation of new blood cellular components in the bone marrow.
Okay, there's one external dietary peptide mentioned in the research that I really want to unpack here. Lactotripeptides.
Ah, yes. The milk peptides.
Exactly, naturally derived from milk. The text notes that while they might reduce blood pressure, the scientific evidence in human trials is frustratingly mixed.
Very mixed.
But if our analytical technology is sophisticated enough to map the mirrored geometry of platypus venom down to the individual atom, why is it so difficult to definitively prove whether a simple 3-amino acid chain from milk lowers blood pressure?
The great question. And the difficulty lies in the difference between studying a molecule in a sterile petri dish versus studying it inside a living, breathing human.
Because it's chaotic.
Exactly. As we established earlier, the human body is a deafening symphony of peptide signaling. At any given millisecond, millions of overlapping messages are firing to raise pressure, lower pressure, manage stress, balance electrolytes.
So if you isolate a lactotripeptide in a vacuum, you can clearly see the chemical mechanism where it inhibits an enzyme that constricts blood vessels.
Right, the mechanism works in isolation.
But when a human actually drinks it, that tiny 3-amino acid chain gets thrown into a biological hurricane.
A literal hurricane, yes.
It gets degraded by aggressive stomach acids. It has to absorb through the intestinal wall, and then it has to compete with thousands of louder systemic signals just to be heard.
Exactly. So the mixed evidence doesn't necessarily mean the mechanism is fake. It just exposes the absolute limits of isolating one variable in an unimaginably complex system.
Right.
You cannot decode the plot of a massive novel by analyzing a single 3-letter word out of context. The systemic noise just drowns out the isolated mechanism.
Which perfectly transitions us to the modern frontier of this science. Because knowing how depthly these tiny chains control our biology, researchers are no longer content just observing them or extracting them from milk.
No, we are far past that.
They're actively engineering them from scratch. And the research extensively covers the development of self-assembling peptides.
Yes. This is where biology completely merges with material science. Scientists are synthesizing specific amino acid sequences engineered to automatically organize themselves into larger, highly complex nanostructures without any external guidance.
Let's focus on the mechanism here, specifically regarding peptide dendrimers, because these are described as highly branched, tree-like molecular structures. How does a linear chain of synthetic text messages suddenly decide to build itself into a microscopic tree?
It relies entirely on the chemical personalities of the amino acids used.
Personalities.
Yeah. Some amino acids are strongly hydrophobic. They aggressively repel water. Others are hydrophilic. They are chemically attracted to water.
Okay, so it's a push and pull.
Exactly.
Okay.
By meticulously sequencing a peptide with alternating water-loving and water-hating sections, scientists can drop that peptide into a fluid environment like the human bloodstream.
And the instant it hits the water, the hydrophobic sections violently tuck inward to hide, while the hydrophilic sections pull outward.
Yes.
And that violent push and pull forces the entire structure to snap into a specific, predetermined 3D shape, almost like a microscopic cage.
A cage is a great way to picture it. And that hollow cage can be loaded with therapeutic payloads,
Like what?
like fragile genetic material or highly toxic chemotherapy drugs, encapsulating them safely until they reach the target tissue.
Man, that structural delivery system connects flawlessly to the next major engineering breakthrough mentioned, cell-penetrating peptides.
Yes, there are Trojan horses.
Right, because the loud bilayer of a human cell is this incredibly strict, heavily guarded barrier. Its entire evolutionary purpose is to keep the outside world out.
It is a fortress. And traditionally, getting a large drug molecule inside a diseased cell required utilizing brute force, which often just destroyed the cell in the process.
But cell-penetrating peptides are sequences specifically engineered with a high concentration of positively charged amino acids.
Yes, positive charges.
And since the surface of a cell membrane carries a negative charge, the peptide essentially acts as a molecular skeleton key. It binds to the negative charge, subtly reorganizes the lipids, and physically slips right through the barrier without lysing or breaking the cell.
It's incredible. By covalently attaching a therapeutic drug to one of these cell-penetrating peptides, modern medicine has essentially created a biological Trojan horse.
We can sneak right past the guards?
Exactly.
Yeah.
We can now bypass the body's natural defenses to deliver highly targeted treatments directly into the cytoplasm or even the nucleus of a diseased cell, leaving all the surrounding healthy tissue completely untouched.
Wow. And finally, we have peptidergic agents.
Yes.
These are synthetic drugs explicitly designed to directly modulate the peptide systems already running in the body. For instance, opioidergics are designed to target the opioid neuropeptide system.
Right.
So instead of flooding the body with an external blunt force chemical to mask a symptom, these drugs are architected to fluently speak the body's native peptide language.
They interact directly with the receptors to upregulate or downregulate the actual text messages your brain is sending regarding pain, mood, or inflammation.
It's so elegant.
Really represents a massive paradigm shift from blunt chemical intervention to precision molecular modulation.
It fundamentally changes how you view the machinery of life. I mean, we started this journey by looking at the simplest 2 to 50 amino acid chains, the short, hyperspecific text messages of biology.
We did.
We saw how evolutionary pressure forced microbes to build non-ribosomal artisan workshops, turning out cyclic hybrid weapons to survive in the dirt.
We explored the extremes of post-translational modification, from the mere D-amino acids in platypus venom that render it invisible to our immune system, to the highly efficient salvaged peptone chunks derived from digesting larger proteins.
And we saw how these functional families run our own internal networks, culminating in a future where we leverage chemical affinities to fold dendrimers into tiny cages and utilize positive charges to slip biological Trojan horses directly through cell walls.
The core realization here is profound. I mean, the next time you experience a sudden shift in mood, a spike in hunger, a response to a breakthrough medication, or even the sharp sting of venom, you'll understand the hidden mechanism at play.
It's peptides all the way down.
A microscopic, perfectly ordered chain of peptides is quietly executing that command behind the scenes.
It is the invisible scaffolding of biology. And it leaves me with one final question to consider.
What's that?
Well, we know that a single geometric modification, just flipping an amino acid left to right, can turn a benign biological chain into an undetectable stealth weapon.
Right, the platypus venom.
Yeah. So, as our engineering capabilities advance to the point where we are writing our own synthetic peptide sequences from scratch, what completely undiscovered, highly complex text messages might we soon be able to write, fold, and send directly into the operating system of our own DNA?
That is the exact frontier researchers are standing on today.